Hi everyone, welcome to today's lecture. This is designed to bridge the gap between mitosis and meiosis and wrap up some important information about mitosis. What you're looking at in this image is a colorized image of two cells undergoing mitosis. And these are cells from the famous Gila line from Henrietta Lacks, who you may have read or seen the movie about her life. And these cells have been photographed as they are undergoing the process of mitosis. The one in the upper left, you can see this chromosome starting to be pulled apart, so it's in anaphase, and the one in the lower right looks to be in early metaphase, where the chromosomes are kind of aligning in the middle. All right. So I want to review again mitosis. And so as we think about the whole cell cycle, we have interphase that consists of the G1, S, and G2 phases, and then mitosis, or the M phase. And the mitosis is the part where the cells divide. But let's look at that in a little bit more detail here. Okay, so you have interphase, where the chromosomes are uncondensed. They're constantly being transcribed, and then that is translated. So this is not part of mitosis. We formally get into mitosis when we enter prophase, and in prophase, the sister chromatids have been replicated, so the DNA is replicated, and the chromosomes are starting to condense, the nuclear envelope is breaking down, and the spindle apparatus is starting to form. And at the top you can see the illustration of this, and below you can see a photograph of this going on. Here we have prometaphase, and in prometaphase, the chromosomes are starting to connect to the spindle fibers at their kinetochores, and that will be then the beginning of them being able to be aligned, which happens during metaphase. And in metaphase, the chromosomes align on the metaphase plate individually during mitosis. The next step is anaphase, and in anaphase, the chromosomes are pulled apart, the sister chromatids separate, and begin to move toward opposite poles. Telophase, the chromosomes have reached the opposite poles and the nuclear envelope starts to reform. And then cytokinesis technically is the division of the cell into two cells, and you can see that in the first illustration shown there, that the bridge of fibers is starting to constrict around the center of the cell, and then in the second illustration, the two cells have completely divided. All right, so these are those illustrations. And I want to then quickly contrast that to meiosis, because now we've started to dive into meiosis a little bit. So remember, in mitosis, in prophase, the chromosomes condense, but they stay as on their own. Whereas in meiosis, in prophase 1, as you see here, chromosome 1, the two homologous chromosome 1s will join up and create a tetrad, and they will sometimes have crossover. And so this occurs in prophase 1. Then you go to metaphase of mitosis, and you can see that those chromosomes have aligned individually. Whereas in metaphase 1 of meiosis 1, you see that those chromosomes are lined up as a homologous pair. So in anaphase of mitosis, the sister chromatids separate, and in anaphase of meiosis, the homologous pair separates. And if you look at this, then you'll see that you went from two chromosomes here in mitosis before anaphase to two chromosomes after anaphase. So in meiosis, the cell has gone from two chromosomes down to one at the end of meiosis 1. So it's gone from diploid to haploid. In mitosis, it has stayed as a diploid cell. In meiosis 1, it goes from diploid to haploid. And then in meiosis 2, those sister chromatids separate, and remains as haploid cells, each having one chromosome in the case as it's illustrated here. Okay, so that was a brief interlude to take us back to think about the contrast of mitosis and meiosis. Now I want to go back and talk about how cells actually divide and how anaphase happens. So anaphase is really important. You can draw it and just think, oh yeah, the chromosomes separate. But how really do they separate is the question. And I talked about this a little bit last Thursday. And what we find is that the chromosomes are actually pulled apart by a little thing called the kinetochore motor that's attached to the centromere section of the chromosome. That kinetochore motor, there will be one on each of the sister chromatids, and it pulls its way down that microtubule spindle fiber by breaking off little tubulin subunits. Okay, so it is working its way down that that fiber and will eventually make it to the pole of the cell, and that's how the chromosomes are separated. So remember, there's one of these on each of the sister chromatids, and they then end up pulling in opposite directions and separating the sister chromatids. All right. So again, this is in anaphase. The sister chromatids are separated, and that's pulled by the kinetochore. The kinetochores are actually depolymerizing the microtubules, breaking them down, and then reattaching further down the microtubule each time. The microtubule and spindle fiber are the same thing. Right? So here we can see anaphase happening. Chromosomes have been pulled apart. And then just the quick definitions of mitosis and cytokinesis. Mitosis technically is the So here we can see anaphase going on, and in anaphase, the chromosomes are pulling themselves along those spindle fibers that are illustrated in green here. So the kinetochore is depolymerizing the tubulin subunits. Okay, so this is the key part of the movement of chromosomes in mitosis. So as we go on to look at what happens when you have problems in mitosis, we want to remember this and think about how we might address this issue. So cell cycle errors will lead to cancer. And so when we have errors where cells start to undergo mitosis more frequently than they should, it can cause the development of an unwanted growth in the body, and that can lead to cancer. And if we look at cancer rates, and we're going to look first at cancer rates in males, we can see that certain cancers were very prevalent, such as stomach cancer in 1930, and that has declined over time. But other cancers, like lung cancer and bronchial cancer, have increased since the 1930s till peaking in around 1990 or 1995, and now beginning to decrease in terms of the number of deaths that they cause. But this is one, if you see at its peak, about 90 out of 100,000 people, men, died of lung cancer. So about 1 out of 1,000 people, men, died of lung cancer. So now we can look at cancer rates in females, and you can see that lung and bronchial cancer has increased there to about 40 out of 100,000. So not as high as it is in males, but it also has not shown the decline that it has shown in males. Now breast cancer, the death rate has decreased a little bit from around 40 to around 30 per 100,000. But you can see a very different suite of cancers here. The reason for the decline in stomach cancer is probably linked to change in the way that food was packaged and the elimination of certain types of metals in food canning. So you can see that in both males and in females that stomach cancer has declined. Deaths caused by that. There are two main types of cell defects that can lead to cancer. The first one is when cells are producing proteins required for cell growth, and they're active that when they should not be. So instead of a cell deciding it's time not to divide because a space is crowded or there's no need for additional cells, these will actually stay active and cause the cells to divide. And then you have other ones that lead to defects in the that will prevent tumor suppressor genes from shutting down the cell cycle. So there are some tumor suppressor genes that can cause the cell cycle to stop. So what is a tumor? We can start off with tumors is when you have a group of cells in a multicellular organism and it's beginning to divide in uncontrolled fashion. So benign tumors, sometimes people have those, and they're non-invasive and non-cancerous. And then malignant tumors can become invasive, and they spread through the body through the lymph and blood systems. They then can initiate secondary tumors in other parts of the body, and this is when it becomes very problematic for an individual. When cancers detach from the original tumor and then they start to spread through the body, that's called metastasis. So people will say, so-and-so has cancer and it's really sad because it's already begun to metastasize. And that means it's starting to spread to other parts of the body. So a benign tumor, for example, shown here in the orangish cells, is growing around the blood and lymph vessel, but it's not leaching cells into those. A malignant tumor shown here will start to deposit cells into the lymph system or into the blood vessels, and those will move and then eventually can cause new cancers in other parts of the body. And this progression shows the different stages of cancer and how people will say cells are precancerous when they start to show hyperplasia or dysplasia, and then that's when dermatologists will try to remove things that they see in those stages before they can reach the stage of becoming an invasive cancer or even an in situ cancer. Now, BRCA, you may have heard of this. People call it the breast cancer gene. And BRCA1 and BRCA2 are tumor suppressor genes. And what they do is they play a role in DNA repair. So that if you have mutations in your DNA, defects in your DNA, breaks in your DNA, they will repair that. And that prevents the development of these mutations that lead to breast cancer. So BRCA1, if you have a functional one of those, it repairs the DNA normally, and then you don't have a lot of potential for defective genes then leading to cancer. Right? So again, it provides instructions for making a protein. We always want to think about that what a gene actually does. That protein then will repair those breaks in the DNA, and that can then help prevent cancers from occurring. All right. If you have a defective BRCA1 or BRCA2, for women that increases the likelihood of developing breast cancer. And the same can happen with men, but breast cancer is a pretty rare disease in men. It still does occur. There are genetic tests available for both of these, and so you want to find out if you have a family history of breast cancer, possibly of whether you are a carrier for that, and maybe make decisions about what you would want to do in life. And so that's one of the things that genetic testing has allowed us to start to address. So again, cancer is due to a loss of control in the cell cycle. And the cell cycle is regulated through signals in the cells. And if those signals are present, but the cells don't respond to them, that's what leads to cancer. So cancer cells will be exposed to those mechanisms that are trying to stop cell division, but they don't respond properly. So that's one way that it can occur. It can occur through a lack of what's called density-dependent inhibition. Density-dependent meaning when crowding happens, the cell should stop dividing. And then you can also have anchorage dependence where mammal cells, or sorry, not mammal, where animal cells in general should be anchored to a substrate, anchored to something like a bone or a muscle or a tissue before they divide, whereas cancer cells will divide on their own, floating through the bloodstream or floating through the lymph system. Normally, a cell needs growth factors to divide, but cancer cells can escape that in different ways. Some cancer cells make their own growth factor, and some cancer cells convey the signal that growth factor is present without it being actually there. And again, this all goes back to the cell cycle control system. The cell cycle control system will tell cells not to divide if there's a problem in the cell or if there are too many cells already. And cancerous cells escape that and ignore that cell cycle control system. So we can look here at a model of metastasis with breast cancer. A tumor is developing and the tumor expands, and eventually that tumor expands and starts to release cancerous cells into the blood vessels and in the lymph system. And then those can move to other parts of the body. So in women, when they discover breast cancer and they remove the tumor, they typically will remove lymph nodes along the arms and along the sides of the chest to determine if there are cancerous cells in those lymph nodes. And if there are, that changes the type of treatment that they're going to do and how aggressive they need to be, because the cancer in that case has the potential to metastasize. So I want to talk a little bit now about cancer chemotherapy. So many of you are familiar with the idea that when somebody has cancer, then they will begin to undergo chemotherapy after it may be in combination with surgery to remove the tumor. It may be in place of surgery to remove the tumor. There are a couple drugs that are important drugs in this that were derived from plant chemicals. So plants have secondary chemicals that may make them unpalatable for things to eat them, or may make them able to grow in certain conditions. And those have been found to have potential for use in cancer treatment. So Vincristine comes from a plant called the Madagascar periwinkle, shown here in the lower corner. And that Madagascar periwinkle has an alkaloid in it that's called vincristine. And it's a mitotic inhibitor, so it stops mitosis. And the way it does it, let's go back and think about our mitotic spindle that's all these little tubulin subunits built up into microtubules. Well, vincristine will bind to that tubulin before it can be built into microtubules. And so without microtubules, you never get to the stage of having a mitotic spindle apparatus. So again, vincristine is a mitotic inhibitor. It binds to tubulin, those little tiny subunits, and it prevents the assembly of microtubules that are necessary for mitosis. A different drug derived from a tree called the Pacific yew is, and here's a yew berries and the vegetation. It's an evergreen found in the Pacific Northwest. And it's derived from the bark of that. And what it does is kind of the opposite. Once the microtubules have formed, it attaches to them and prevents them from being broken down. So that kinetochore motor that we talked about earlier that normally breaks down the microtubules, depolymerizes it, and moves its way along the microtubule is unable to do that because Paclitaxel or Taxol has stabilized that microtubule. So this process where microtubules are broken down by the tubulin subunits being broken off does not happen. So if we look at these kind of in a review, vincristine prevents microtubule assembly, Paclitaxel prevents microtubule breakdown. So one stops the microtubules from ever being formed, the other stops them from being broken down once they're formed. Both have the effect of stopping mitosis. One does it before you can get to the stage of prometaphase because you can't develop microtubules to attach to the chromosomes, and the other does it between metaphase and anaphase, so anaphase can't happen, the chromosomes can't be pulled. There are other drugs, cancer chemotherapy drugs, that act in different ways. So methotrexate is not derived from a plant. It's a chemically synthesized drug, and it acts during the cell cycle, during the S phase, and it prevents DNA and RNA synthesis. So in this case, cells can't replicate because they can't replicate the DNA, and so it stops mitosis from ever happening because there's no replicated chromosomes. So these are different ways that the cancer can be treated. And just a few years ago, they released a new drug for cancer treatment. It's called Kadcyla, and it's actually not completely new. It takes two drugs, trastuzumab and DM1, and combines them. And trastuzumab is a targeting drug. So this it's a monoclonal antibody drug that allows them to target where the cancer treatment will go. And then DM1 is a microtubule assembly inhibitor. So similar to Vincristine, it prevents microtubules from being assembled. Now, it's a revolutionary new drug because it allows mitosis to continue to go on in parts of the body that aren't targeted, but the targeted cells around the cancer then are where mitosis will be stopped because the DM1 will stop microtubule assembly. It costs $8,900 per month or $94,000 for a 9.5-month treatment. So it is an incredibly expensive drug, but it does provide great hope in being able to treat cancers effectively. Now you think about that and you think about how much drugs do cost, and the cost of drug development has really increased substantially. And so along with that has the cost of drugs that make it to market. And for every drug that makes it to market, there's about $2.5 billion spent on that drug before it makes it to market. And so they need to recoup that research and investment. But it still is pretty alarming how expensive that is. Some drugs are $100,000 per treatment, but have $2.6 billion in development cost. You can see that they would need to sell quite a few of those drugs to even make up the development costs. So another drug that allows the treatment of hepatitis C is called Sovaldi, and that costs $1,000 a pill, and you take that for 12 weeks, and it's $84,000 for the full regimen. And this has really impacted things like the state Medicaid plans and so on. The university medical plan, for example, has had a lot of challenges in being able to pay for this drug for people in the community that need it. There's other drugs that are very expensive, things like Abilify. Those are things that cost $1,000 a month, and those are taken for years. And so drug costs are very expensive and very challenging, and bring into question a lot of things that hopefully you'll have a chance to talk about in some of your HPS courses, like why we have for-profit medicine in general and what the pluses and minuses of for-profit medicine are.